In the second year botany, if you are seeing the long answers, that is 8 marks questions, mainly 8 marks questions, important questions if you are discussing. The important questions mainly consisting of cycles mainly, Calvin cycle, glycolysis, Krebs cycle. These three are from the first unit, that is the transport in plants. Then after that thing, if you are observing, from the genetics, there may be chance of asking the dihybrid cross or independent assortment, or else from the last unit, that is actually biotechnology. In the biotechnology, there may be chance of asking the steps for the construction of rDNA and then tools of rDNA technology. Then lastly about the C-waste treatment. These are the important questions, important 8 marks questions. Now we will discuss about the quick revision of all this, revision of the points of these answers particularly. First of all, if you are considering about the Calvin cycle we are calling it as a dark cycle or carbon dioxide fixation cycle. Calvin cycle has been proposed by the Melvin Calvin, and the Calvin cycle, if you are seeing particularly, Calvin cycle is mainly consisting of 3 steps: carboxylation, reduction, regeneration. Carboxylation is a step, particularly it is consisting of fixation of carbon dioxide. In the carboxylation step of the Calvin cycle, ribulose 1,5-bisphosphate is a carbon dioxide acceptor in the presence of enzyme rubisco. Rubisco, it means ribulose 1,5-bisphosphate carboxylase. Then this ribulose 1,5-bisphosphate, which is a 5-carbon compound, which is combining with the carbon dioxide, 5 plus 1, 6 carbons. Then the compounds which are forming 12 PGA, 12 phosphoglyceric acids are formed by the first reaction, that is the carboxylation. The 12 PGA which are formed here particularly, they are undergoing the reduction. That is the second step of the Calvin cycle. The 12 PGA, they are undergoing the reduction. These 12 phosphoglyceric acids which we had, they are undergoing the reduction. They are forming first the 1,3-bis phosphoglyceric acid in the presence of phosphoglycerokinase enzyme. This 1,3-bis phosphoglyceric acid which they are forming here 12. This 12 moles of 1,3-bis phosphoglyceric acid, again they are reduced to 12 G3P, 12 glyceroldehyde 3-phosphate in the presence of enzyme G3P dehydrogenase. This is the second step, is the reduction. Lastly, what we got now by the reduction? By the reduction we got the 12 G3P. In the Calvin cycle, in the regeneration step, we are not using the 12 G3P, but from the 12 G3P mostly we are using 6 G3P. In the very first step of the regeneration, what we are doing, the G3P is combining with the DHAP. Why DHAP is coming here? It means DHAP is the isomerase of G3P. 4 G3P are converted into DHAP. DHAP, it means dihydroxyacetone phosphate. 1 G3P plus DHAP gives rise to what? Fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate again converted into fructose 6-phosphate in the presence of enzyme fructose 1,6-bisphosphatase. This fructose 6-phosphate is combining with the G3P, and then 6-carbon compound fructose 6-phosphate plus G3P 3-carbon compound, 6 plus 3 9-carbon compound. Then in the next step also we should get the 9-carbon compound. The 9-carbon compounds which we are getting here particularly, they are xylulose 5-phosphate and erythrose 4-phosphate. Here erythrose 4-phosphate which we got here, erythrose 4-phosphate is again reacting with the DHAP. This DHAP plus erythrose 4-phosphate, 4 plus 3, 7-carbon compound we should get. The 7-carbon compound is actually pseudoepiclose 1,7-bisphosphate. This pseudoepiclose 1,7-bisphosphate, it is combining with again G3P. 7-carbon compound pseudoepiclose 1,7-bisphosphate plus G3P, 7 plus 3, 10. 10-carbon compound we should get. The 10-carbon compound which we are getting here is particularly xylulose 5-phosphate and then ribulose 5-phosphate, ribose 5-phosphate. 5 plus 5, 10. Now the xylulose 5-phosphate which we had particularly, xylulose 5-phosphate, 4 moles of xylulose 5-phosphate in the presence of enzyme xylulose 5-phosphate epimerase, or else ribulose 5-phosphate epimerase enzyme is converting the xylulose 5-phosphate into 4 moles of ribulose 5-phosphate. Then the ribose 5-phosphate which we had particularly, this ribose 5-phosphate in the presence of enzyme ribulose 5-phosphate isomerase, it is converted into 2 moles of ribulose 1,5-bisphosphate. Lastly, what we are getting now, 4 plus 2, 6 moles of ribulose 1,5-bisphosphate we are getting. This ribulose 1,5-bisphosphate which we are getting lastly, 6 moles of ribulose 1,5-bisphosphate, ribulose bisphosphate is converted into ribulose 1,5-bisphosphate. That is the last step. That means in the regeneration what we are getting with the help of the G3P, DHAP, lastly we are getting 6 moles of ribulose 1,5 bisphosphate. Why we are getting the 6 moles? It means here Calvin cycles which we had, for the fixation of 1 carbon dioxide we require 3 ATP and 2 NADH. For the fixation of the 6 carbon dioxide, how many ATP we require? 18 ATP we require and 12 NADPH we require here. This is a Calvin cycle. Remember this point here. The Calvin cycle particularly we have to see the 3 reactions, that is carboxylation, reduction, regeneration. Just we have to see the balancing of these compounds. If you are seeing the balancing of these compounds, then we can write the cycle easily. In the first step, carboxylation, simple, acceptance of carbon dioxide molecule by the ribulose 1,5 bisphosphate. Second step, 12 PGA which is formed here. As the first stable compound which is formed here, it is a PGA, phosphoglyceric acid 3 carbon compound. Hence we are calling this cycle as a C3 cycle. This 12 PGA is reduced to 1,3 bis phosphoglyceric acid. 1,3 bis phosphoglyceric acid is reduced to the G3P. There is a reduction step. In the regeneration step, G3P plus DHAP which we had particularly these two, they are forming fructose 1,6 bisphosphate, fructose 1,6 bisphosphate to fructose 6 phosphate, fructose 6 phosphate plus G3P particularly. It is what we are getting the next compounds: erythrose 5 phosphate, xylulose 4 phosphate, xylulose 4 phosphate plus DHAP, pseudoepiplose 1,7 bisphosphate, pseudoepiplose 1,7 bisphosphate to pseudoepiplose 7 phosphate, pseudoepiplose 7 phosphate plus G3P, that we are getting the 10 carbon compound next, that is the xylulose 5 phosphate, then ribose 5 phosphate, xylulose 5 phosphate, ribose 5 phosphate. Both are combining, lastly they are forming the ribulose 1,5 bisphosphate 6 moles. This is the Calvin cycle. These all steps we have to remember, right? The next, if you are discussing about the glycolysis, very important for the 8 marks glycolysis also. Glycolysis is also known as EMP pathway. EMP pathway, it means Embedon, Mayrhof and Paranas pathway. Glycolysis simply it means conversion of 1 mole of glucose to 2 moles of pyruvic acid. That means 6 carbon compound glucose we are converting into 3 carbon compound pyruvic acid. That's why we are getting 2 moles. Right? Now, in the glycolysis, what is happening here? In the glycolysis, first what is happening here? Glucose, first it is converted into glucose 6 phosphate in the presence of enzyme hexokinase. Here 1 ATP is utilized. Now glucose 6 phosphate which we had particularly, it is again converted into fructose 6 phosphate in the presence of enzyme hexose phosphate isomerase. Fructose 6 phosphate which we had particularly, it is converted into fructose 1,6 bisphosphate. Here also ATP is utilized in the presence of enzyme phosphofructokinase. Fructose 1,6 bisphosphate which we had particularly, it is again converted into what? In the presence of enzyme aldolase, it is splitted into G3P plus DHAP. Now the triose compounds which we are getting G3P plus DHAP, they are undergoing the isomerization, and now the G3P which we had particularly, G3P is converted into what? 1,3 bis phosphoglyceric acid in the presence of enzyme G3P dehydrogenase. Now 1,3 bis phosphoglyceric acid which we had particularly, now it is converted into what? 3 PGA in the presence of enzyme phosphoglycerokinase. This conversion of 1,3 bis phosphoglyceric acid to 3 PGA is a very important step, because this step is known as the first level of substrate level phosphorylation. Here ATP is formed here. Now whatever the 3 PGA which is formed here, it is converted into 2 PGA in the presence of enzyme phosphoglyceromutase, and 2 PGA is converted into phosphoenol pyruvate. Now the phosphoenol pyruvate is which we had particularly, it is converted into what? Pyruvic acid in the presence of enzyme enolase. Here also conversion of phosphoenol pyruvate to pyruvic acid, it also includes the second level of substrate level phosphorylation, that is the formation of ATP. Lastly 2 moles of pyruvic acid we are getting, and here particularly remember the point, at the end of glycolysis particularly, net gain of the ATP is a 2 moles of ATP particularly. 2 are utilized and 4 are produced. Net gain we are getting actually particularly 2 moles of ATP. This is the glycolysis. These steps we have to remember compulsory. Then if you are seeing the Krebs cycle particularly, Krebs cycle is also known as Krebs cycle, and then tricarboxylic acid cycle, TCA cycle, or else citric acid cycle. The Krebs cycle which we had particularly, it includes the different steps in the Krebs cycle. Krebs cycle always starts with the condensation reaction. That means acetyl coenzyme A plus oxaloacetic acid. Acetyl coenzyme A in the Krebs cycle which we are getting particularly, we are getting it by the pyruvic acid. Pyruvic acid which is formed in the glycolysis, this pyruvic acid is lastly 3 carbon compound is giving rise to 2 carbon compound, that is acetyl coenzyme in the presence of enzyme pyruvate decarboxylase. Acetyl coenzyme A plus oxaloacetic acid, they are combining both. This reaction we are calling as a condensation reaction. Then 4 carbon compound oxaloacetic acid plus 2 carbon compound acetyl coenzyme A, they forms the citric acid. In the presence of enzyme citrate synthase. Now citric acid which we got here particularly, citric acid is converted into isocitric acid. This is the dehydration reaction. And this isocitric acid particularly which we are getting here, that means aconitase we are getting particularly in the presence of enzyme aconitase, hydration, and aconitase which we had particularly, this aconitase is converted into isocitric acid. This is a dehydration reaction, removal of the water. Isocitric acid which we got here particularly, it is converted into what? Oxalosuccinic acid. This oxalosuccinic acid which we had particularly, it is converted into what? It is converted into alpha ketoglutric acid. The alpha ketoglutric acid is converted into succinyl coenzyme A. This succinyl coenzyme A is converted into succinic acid. Succinic acid is converted into what? Fumaric acid. Fumaric acid is converted into malic acid. Lastly, malic acid gives rise to oxaloacetic acid. This is the cycle of the Krebs cycle, and remember the point here, in the Krebs cycle particularly, condensation reactions, hydration reactions, dehydration reactions, oxidation reactions are taking place here. 4 oxidation reactions takes place in the particular Krebs cycle. And this oxidation reaction, it means there we are getting the enzyme as a dehydrogenase. Remember the simple point. In the complete Krebs cycle, in the conversion of the succinyl coenzyme A to succinic acid, we are getting the ATP or GTP here. Rest of the things we are getting from the fumaric acid to malic acid, we are getting FADH2, and then NADH we are getting. Lastly, by the Krebs cycle, what is happening here? Last product we are getting, there is oxaloacetic acid from the malic acid here. And in the Krebs cycle, we have to remember reactions, we have to clearly write about the enzymes and what are the products we are getting, all these things in the Krebs cycle particularly. Krebs cycle is also very important for the 8 marks here. And remember the point, these 3 cycles are very important. Calvin cycle, glycolysis, Krebs cycle. From these 3 compulsory, we will get 1 question in the examination. Just we have to see the products which we are getting, what are the enzymes, we have to see the steps in particular things. Flowchart is very important, plus reactions also we have to write here particularly in these 3 cycles. This is about the 3 cycles of the first unit. If you are moving for the second unit, actually there may be chance of asking law of independent assortment or dihybrid cross for the 8 marks. Dihybrid cross or law of independent assortment mainly consisting of what? Round yellow and wrinkled green. The both characters which we had particular, that means 2 characters we are crossing. 4 alleles particularly we are crossing. Lastly we are getting the 16 alleles we are getting in the dihybrid cross. In the dihybrid cross, we know about the Punnett square particularly Punnett square, and we are crossing the 2 characters. Lastly we are getting 16 alleles we are getting particularly. This 16 alleles which we had particularly, this 16 alleles we have to take the phenotypic ratio, genotypic ratio. By the dihybrid cross, phenotypic ratio we are getting 9:3:3:1, and then genotypic ratio we are getting 1:2:2:1:2:1, like that we are getting here. That complete thing we have to do, and don't confuse in this particular question that independent assortment and then dihybrid cross both are same. Independent assortment we are calling particularly dihybrid cross because in this phenomena particularly what is happening, it means round yellow then wrinkled green. These 2 characters of a seed characters we are saying, that means round is the shape of a seed and yellow is the color of the seed coat, wrinkled is the shape of a seed, green is the color of the seed coat. These 4 characters we are crossing, and as we are crossing this 4 characters, what characters we should get? Actually same characters we should get, but new characters we are getting in the next in the second generation, in the F2 generation. That's why we are calling it as a law of independent assortments, right? Then this is in regarding the second unit. And the biotechnology unit, in the biotechnology unit actually very important steps involved in the formation of rDNA. rDNA, it means recombinant DNA. Important steps which are involved in the recombinant DNA actually, first of all, isolation of the desired DNA we are saying. In the isolation of the desired DNA particularly, it is the first step. The second step will be isolation of the desired DNA is the first step. Then second step will be separation of a DNA. Then third step will be ligation of the isolated DNA to the suitable vector. Then fourth step will be insertion of the recombinant DNA which we got here into a suitable host. Then the next step will be replication in the suitable host, and the next step will be replication. After replication, what is happening actually? Selection of the recombinant DNA in the particular host. Lastly, we are seeing about the identification of a desired DNA, desired recombinant DNA. Lastly, downstream processing we are saying. Mainly these steps we had in the recombinant DNA. First of all, the first step as we are discussing, isolation of a desired DNA. Isolation of a desired DNA or suitable DNA, it means first what is happening here particularly, it means we have to isolate, we have to select the DNA which we require. This is done by what? Actually this is done mainly by the particularly by the selectable markers, by the restriction enzymes, all these things here. That means from a DNA sequence we are selecting the DNA sequence which we require to form the recombinant DNA. For this thing what we are doing, centrifugation technique we are doing, and we are using the lysozyme enzyme. This lysozyme enzyme always what it is doing actually, it is disintegrating the cell wall of the particular cell. Whenever the disintegration of the cell wall takes place, then what will happen? We are going particularly there is an easy way for the isolation of the DNA, and this isolation of DNA consisting of different polyglycol method. All these methods we are seeing in this isolation of the DNA. Then second step of the isolation of the DNA, second step particularly. Separation of the DNA we are saying. So DNA we have been isolated, and separation of the DNA we are saying particularly, or else selection of the suitable marker or suitable vector we are saying. Before that actually separation of the DNA we are saying. That means many DNA which we are isolated. From that isolated DNA, first the bunch of DNA we have to separate the DNA. Separation of the DNA mainly consisting of a agarose gel electrophoresis technique. Agarose gel electrophoresis technique mainly consisting of a agarose gel plate wells we had. Then we are dropping the desired DNA sample, and we are connecting it to the cathode and anode. By connection of the cathode and anode by the gel electrophoresis technique, what is happening here? DNA separation takes place, and remember the point particularly, DNA is always negatively charged. DNA moves towards the positive charge. By the gel electrophoresis method, DNA bands has been separated. By the particular ultraviolet radiation technique, we can identify the bands of the DNA. By separation we can do here. Next, selection of the suitable vector. Suitable vector we have to select, because actually we have to insert the isolated DNA into the host now. With the help of the suitable vector only we are inserting. Suitable vector particularly we are selecting it as a plasmid only. Plasmid is a particular suitable vector for the isolation of the DNA. That means for the DNA to take the plasmid particular to insert into the suitable host here. Plasmid is suitable vector. Vector always carries the isolated desired DNA to the particular host here, and here suitable vector will be consisting of a vector DNA plus the isolated DNA both. Whenever we are ligating, whenever we are combining the particular isolated desired DNA plus vector DNA, then what we are saying? We are saying particularly it as a recombinant DNA, because two DNA we are combining as by the ligation. That means DNA ligase is an enzyme which are combining these two, and this rDNA which we got here particularly, we are inserting this rDNA into the suitable host. Into a suitable host particularly by the different methods actually, heat shock, and then by the heating methods, and the heat shock method, by the bombardment, ballistic method, all these methods, and shotgun methods, we are inserting the particular rDNA into the suitable host here. In the suitable host, what is happening? Replication takes place particularly. After the replication, what is happening here? We have to select the replicated rDNA. Why we are doing the replication? It means from the small copies, from the one copy of rDNA, we require many copies. In a less time, we require more amount of copies. That's why we are doing the replication suitable DNA. Suitable vector particularly, or suitable host particularly. Suitable host, the replication takes place. After replication, again we are selecting the desired rDNA, desired gene of interest particularly, foreign DNA particularly, by what? By the different methods. Different methods mainly includes what? Ampicillin resistant particular isolated desired rDNA we are selecting. That means whenever we are growing this rDNAs which we got from the host here, the rDNAs which are capable of growing in the ampicillin medium particularly, then they are known as rDNA particularly, which we require, desired rDNA. After separation of this desired rDNA and desired rDNA, we can also identify by the blue-white selection method also. After separation of this desired rDNA, next step we are including, there is a downstream processing. Downstream processing includes the purification, selection and purification. Lastly, there is the last step here. This is about the steps involved in the formation of RDNA. Isolation of a desired gene, then selection of the suitable, then the insertion of a desired gene particularly into a suitable vector, then selection of suitable vector, then separation of the particular desired gene DNA by the gel electrophoresis, then insertion of a particular desired DNA into the suitable vector, that is the formation of rDNA, and after the formation of rDNA, insertion into suitable host cell. After suitable host cell, insertion into suitable host cell, replication. After replication, then we are selecting the desired for DNA rDNA which we require. Lastly, downstream processing we are doing. This is the rDNA steps involved particularly. Then tools which are involved in the rDNA methods mainly consisting of what particular restriction enzymes we are saying. In the restriction enzymes, exonuclease we had, endonuclease we had. Exonuclease always cuts at the ends. Endonuclease always cuts in the inside of the DNA sequences particularly. Then particularly cloning vectors we had. In the cloning vectors particularly suitable sites, origin of replication, selectable markers, resistant genes, ampicillin resistant gene, tetracycline resistant gene, all these things we are seeing in a particularly these cloning vectors. Examples of the cloning vectors, if you are taking particularly PBR 322, that means plasmid, Bolivar rotary gauge method, rotary gauge plasmid. Then PUC 119, that means plasmid, University of California. Different things we have seen in the cloning vectors. After the cloning vectors, many things we have seen. Selectable markers, all these things we are seeing. Then these things we are seeing in particularly tools of rDNA. This may be, there may be chance of coming this for the 8 marks also. This is regarding the biotechnology. After this biotechnology, particularly if you are seeing after this biotechnology, then we can see in the last chapter, that is actually sewage treatment with the help of the microorganisms. In the sewage treatment of the microorganisms, particularly 2 steps we had: primary treatment, secondary treatment. Primary treatment mainly consisting of basic steps, that is actually collecting of this sludge, that means formation of the primary sludge is a particular thing in the primary treatment. That means whenever we are moving the effluents in a particular thing, actually what is happening, the debris, the solid wastage which we have, the solid wastage is collected in the primary treatment. There is a primary sludge we are saying. And after the primary treatment, the effluent which we are getting, particularly that effluent we are treating in the secondary treatment. In the secondary treatment, we have different steps actually. In the secondary treatments, aeration we had, effluation, aeration, and then we are treating the particularly the effluent with the aerobic bacteria, anaerobic bacteria. Lastly, we are getting the biogas with the help of the methane and then the sulfide, hydrogen sulfide. We are getting the biogas. That these steps are involved in the microbes involved in the sewage treatment. 2 steps mainly we had in this method: primary treatment, secondary treatment. In the secondary treatment, mainly BOD, biological oxygen demand, also very important in the secondary treatment. We have tried about this thing. BOD, whenever the BOD level is increasing, it means the contamination is increasing, we are saying particularly. And these are regarding the long answers of the botany second year in the final examination. Out of the 3 cycles, Calvin cycle, then glycolysis and Krebs cycle, 1 question we are getting. Then in the second unit, dihybrid cross, from the biotechnology, that means actually process of steps involved in the formation of rDNA, and tools of the recombinant DNA. Out of these 2, there is a chance of getting 1, and compulsory sewage treatment. These are the long answers. Out of 4, we have tried 2 particularly. Just whatever we revised in this quick things, just you have to remember the points. This point should be covered in the answers particularly.